Hello friends, welcome back to my channel Plant Pathology Pathshala. So in this video we will deal with PAMPs, the Pathogen-Associated Molecular Patterns. So we will deal with the definition, who discovered it, it's about its discovery, then about the types of PAMPs that are present in different organisms, and based on their localization when many types of PAMPs are there, then about its characteristics. We will compare it with the effectors. Okay, so without further delay, let's start the video. So first about the definition. So PAMs are pathogen associated molecular patterns. So these are small molecular signature motifs that are constitutively present in pathogen. See, these are very small in size, very minute, okay, and these are small molecular signature motifs. So these are signature part of the plant, like the flagellin protein. It is a signature molecule for the bacteria as well as for the fungi also. Okay, and if you consider the peptidoglycan molecule, then it will definitely present in a bacteria only. So the peptidoglycan is a signature molecule for the bacteria. It is not present in any type of algae or any fungi. Okay, if a bacterial pathogen is there, then the peptidoglycan PAMs will be produced. Okay, so these are signature molecules that are constitutively present in pathogen. Constitutively present in pathogen means it is present as a structural thing. See, the effectors are not constitutively present. Effectors are induced by the pathogen when it attacks a host. It is not the signature motif. Okay, but here PAMP is the molecular signature motif that is constitutively present. See, if peptidoglycan is not present in bacteria, how it will survive? Because the peptidoglycan is the major constituent of the bacterial cell wall, so it provides protection as well as structural integrity. Everything depends on the peptidoglycan. It's constitutively present in the bacterial pathogen. Okay, that's why PAMs are very important for the pathogen. Okay, for the pathogen survival. So these PAMs are recognized by pattern recognition receptors, PRRs, present in the host. So the PRR, these are the molecule present in the membrane of the host that recognize the signature molecule of the pathogen and induce pattern triggered immunity, PTI. Because this PAMs are a signature pattern of the pathogen. As the immunity is triggered by the PRR by recognizing a particular pattern, it is known as pattern triggered immunity. Okay, so this is basic about the PAMs. We will detailedly discuss in the further slides. Okay, so first about the discovery. So first, the PAMP term was introduced by Charles Janeway in 1990. Then Boller and colleague discovered that the Arabidopsis receptor FLS2 recognizes the bacterial PAMP flagellin FLG22. This is the first PAMP and PRR combination discovered. Okay, remember this. Boller et al. 2000 discovered FLS2, the PRR, and the PAMP molecule first discovered is FLG22, that is a flagellin protein. Okay. Next, in case of fungi, in 2006, CERK1, this is a chitin binding receptor, chitin binding receptor kinase that identified to recognize the fungal chitin. So here, in case of fungi, the PAM molecule first discovered was the chitin molecule, and the PRR that recognizes the PAM molecule discovered was, or the chitin molecule discovered was, the CERK1. Okay, two combination we have studied. For in case of the bacteria, the FLS2 and FLG22 combination, and in case of fungi it is CERK1 and chitin. So remember the examples. Okay, and the PAM molecule was first discovered by Janeway 1990. The term actually discovered by him. So this is in brief about the discovery of PAMPs. So next, about the characteristics of the pathogen-associated molecular patterns. So this molecule are conserved, essential for the pathogen survival, not easily alterable, then recognized by PRRs, the recognition trigger pattern triggered immunity. So conserved means just like I have told you the peptidoglycan example. It is conserved throughout all bacteria. Can you give any example of bacteria that is lacking the peptidoglycan? No, definitely. So it is conserved across the all microbial taxa, means that particular microbial taxa. See, chitin, this is particular for the fungi. Then oomycete contains beta glucan. This viruses contain code protein, double-stranded RNA or DNA or any nucleic acid. So these are the conserved thing. Without that particular molecule, the pathogen can't survive. So it is essential for the survival of the pathogen. Then it is can't be easily alterable, as it is highly conserved, so it can't be alterable. See, we have discussed in effectors that if a particular effector is recognized by our plant R gene, then the pathogen actually modify the effector so that the effector will not recognize by the R gene, and it will trigger effector trigger susceptibility, and disease will be induced or plant becomes susceptible. But here the pathogen cannot alter its PAM molecule, because these are very essential as well as conserved thing for the plant, for the pathogen. So as it is recognized by the PRR molecules and plant is activating the defense, still this pathogen can't change it. It has to deal with that. That's why actually to overcome this defense or this PTI, plant this pathogen produce the effector molecules. So as we have already discussed, it is the PAM molecule that are recognized by the PRR, that are the receptor molecules present on the plant and further deactivate pattern triggered immunity. Then examples of major PAMs present in specific pathogens, like the bacteria, PAMs are FLG22, then EFTU elongation factor, then lipopolysaccharide, peptidoglycan, then fungi, it is chitin, then beta-1,3 glucan, then for the oomycetes it is allicin, very specific for the phytophthora, and beta glucan, then for the virus it's double-stranded RNA or DNA or nucleic acid, and the code protein. Okay, so these examples are important for MCQ based exams. Remember this. And the characteristics is also very important. These are conserved essential molecules that cannot be easily altered, and it is recognized by PRR that ultimately trigger the pattern triggered immunity. Next, about the classification of PAMs. So based on the localization where these PAMs are present, these are classified into two major groups, that are the cell wall or membrane associated PAMs and cytoplasm associated PAMs. So the examples of cell wall or membrane associated PAMs are flagellin, chitin, liposaccharide. Means these are present at the outer region of the pathogen. Flagella is present means outside the cell wall. Then chitin located in the cell wall. Lipopolysaccharide, this is the exopolysaccharide region. Then about the cytoplasm, means it is present in the cytoplasm of the pathogen, like the cold shock protein, then elongation factor. Okay. So this is about the classification of PAMP. Then about the recognition pathway or PTI pathway. So when a pathogen come in contact with a plant, as the PAMPs are the molecular pattern or the signature molecules of the pathogen. So these are definitely present associated with the pathogen. So the PAMPs molecule are then recognized by the receptors, that is the pattern trigger receptor present in the plasma membrane of the plant, that ultimately trigger a basal level of immunity, that is PTI. Okay, and PTI response is associated with ROS burst, maybe Callose deposition, then synthesis of some antimicrobial compounds, then transcriptional reprogramming. Transcriptional reprogramming is it will activate some genes, some gene transcription, so that some new proteins will be, and protein or RNA will be formed. Okay, then activate signaling pathway like MAPK, then calcium dependent protein kinases. So we will discuss the signaling pathway that are regulating the defense mechanism of plant in separate video. And about PTI and PRR also I will make a separate video. In the next video only we will discuss about PRR. What is PRR? The pattern recognition receptor, where it is present, what are the types of PRR present in the plant. See, this combination PRR and PAMPs, these are this interaction lead to PTI, whereas the effector and R gene, this recognition will lead to ETI, effector triggered immunity. Okay, so this PTI is the basal level of immunity and ETI is the second level. Okay, or you can say the advanced level of immunity for the plant. Okay, so this is all about the PAMP molecule. In the next video we will discuss about the pattern recognition receptor, means what are the motifs are there, how it recognizes the PAMP, what are the types, what are the mechanism of recognition. So everything we will discuss. Okay. So this is all about today's video. So thank you for watching.